Hi everyone, welcome back to the Immunology series. Now today we have a very open-ended lecture titled Immune Failure. Now sometimes our immune system fails to fight infections due to having a weakened immune response, and we call that immune deficiency. But immune failure refers to when pathogens develop or evolve ways to evade our immune system. So we are going to look at both bacteria and virus examples of how they find ways to beat our immune response. So let's get started. In this lecture, we are going to look at examples of bacteria antigenic variations and how they cause the immune system to fail to respond properly. Then we will use the influenza virus as the classical example to illustrate antigenic drift and shift in the virus. Lastly, we will describe an immune failure condition that is originated from our adaptive immune system. It is called the original antigenic sin or immune imprinting. The reason for the immune system to fail to respond properly is not always because of a weakened immune system. Sometimes it is because the pathogens are tricky. Pathogens, just like us, exhibit genetic variation in the same way that some of us have brown eyes or blue eyes. Pathogens may express different markers on their cell surface. Those markers are especially important for the development of memory or long-term immunity. But these genetic variations also allow them to evade the immune response and prevent effective long-term immunity. Serotype simply refers to the different strains of bacteria or viral pathogen that express a different set of cell surface markers. The term is derived because antibody-based serological assays are used to define differences between the strains. Markers are proteins or polysaccharides with different structures. The structures are different enough that they are bound and recognized by different antibodies. The mainstay or basis of immunological memory is antibodies directed against surface macromolecules. Different epitopes are recognized by different antibodies with different antigen binding specificity, and therefore infection by one strain will not necessarily yield full protective immunity from other serotypes. Now let's look at a few examples of bacteria antigenic variations and their other ways to prevent us from mounting a proper immune response. One of the most common antigenic variation in bacteria is on the capsular polysaccharides. Now, capsular polysaccharides are carbohydrates that coat the outermost bacterial surface, forming a slime layer. Each serotype or strain expresses different carbohydrates or polysaccharides on its surface. The diversity in the capsular polysaccharides is the source of antigenic variation. There are about 80 to 90 different strains of Streptococcus pneumoniae. It is the common cause of bacterial pneumonia because genetic variation prevents us from developing an effective immunological memory against all strains. Patients must develop a separate immune response to each strain or serotype. It is believed that genetic variation has evolved in Streptococcus pneumoniae as a result of selective pressure exerted by the human immune response. Some capsular polysaccharides are homologous or very similar to ours. This creates a camouflage and shields our immune system from responding. This again ties into the central idea of self and non-self. If it looks like ours, the immune system is not going to react. However, we learned that in the autoimmune lecture that the similarity between pathogen antigens and self-antigens is also referred to as molecular mimicry. and so it may cause an autoimmune response toward endogenous polysaccharides. Now, there are many documented cases linked between infectious disease and autoimmune disorder developments. Another bacteria that commonly cause immune failure is Staphylococcus aureus. Now, it is a gram-positive, round-shaped bacterium that clusters in a grape-like formation. When this bacterium lives on the surface of our skin, they are non-pathogenic and considered to be a part of the normal flora. However, when it infiltrates the tissue, it can cause a wide variety of infection, from mild skin lesions to more serious infections such as pneumonia, meningitis, and urinary tract infection. The most problematic strain of S. aureus is methicillin-resistant Staph aureus, or MRSA, that is commonly associated with hospital-acquired infections. S. aureus and a few other bacteria species evade our immune system by having a capsule. It is a slimy layer consisting of polysaccharides and water surrounding many cells, now also called slime coat, extracellular layer, and etc. Now, difficult to sting as it is, since it is mostly water. Now, this layer protects the bacteria cell and is often associated with pathogenic bacteria because it serves as a barrier against phagocytosis by white blood cells. Encapsulated bacteria are naturally resistant to phagocytosis. The figures shows neutrophil attempting to engulf bacteria but fail without the help of antibodies and complement proteins. After opsonizations by antibodies and complement, the Fc receptor on neutrophils bind to the Fc region of the antibody, and the complement receptors bind to the complement protein. Now, neutrophils can only engulf encapsulated bacteria through this mechanism. Capsular polysaccharides also drive a thymus-independent response. Now remember, T cells normally only respond to processed proteins or small peptides, and not to sugars, carbohydrates, and polysaccharides. Now, T helper cells shown in the figure reminds us that they are normally necessary for full B cell activation and maturation into plasma cells. Now, when encountering capsular polysaccharides, B cells can only be activated with thymus-independent mechanism. These polysaccharides are not as good as eliciting immune response, and not as good as causing memory cells to be formed and isotype switching, so only IgM will be secreted through this process. Now, for children less than 2 years old, the adaptive immune system is not good at responding to thymus-independent response. Mycobacterium tuberculosis is an intracellular rod-shaped bacteria that is the cause of tuberculosis. One of the ways this bacteria evades the immune system is by preventing the fusion of lysosomes with the phagosomes that engulf the bacteria. The bacteria produce a lipid substance that inhibits the fusion between phagosome and lysosome to form the phagolysosome. This allows the bacteria to avoid being killed by the acidic and enzymatic environment of the lysosome and thrive within the phagocyte's vascular system. Listeria monocytogenes is a gram-positive bacillus bacteria. It is a common cause of bacteria food poisoning. One of the ways Listeria monocytogenes avoids detection is by producing a protein called Listeriolysin O, which allows it to escape from the phagosome and enter the cytosol of the host cell. Now, this prevents the bacteria from being degraded by lysosomal enzyme and allows it to replicate within the host cell.
Its name was Enza. I opened the window and into
What you just hear is the roping song. The roping song is a reference to a phenomenon known as St. Vitus dance, a neurological disorder characterized by rapid, irregular, and involuntary movements of the limbs and face, which is associated with the 1918 influenza pandemic. Now, during that time, some patients with severe respiratory symptoms also developed neurological symptoms, including this St. Vitus dance. Now, people with St. Vitus dance would link arms and dance together in a circular motion, singing a song as they went. This behavior was often witnessed in public places such as streets, parks, and hospitals, and was considered a hallmark of the St. Vitus dance associated with the influenza pandemic. Now, so enough with the history. Let's use the influenza virus to explain immune failure in viruses. There are 4 types of influenza viruses, A, B, C, and D. Human influenza A and B viruses cause seasonal epidemics of disease almost every winter in the United States. Influenza type C infections generally cause a mild respiratory illness and are not thought to cause epidemics. Influenza D viruses primarily affect cattle and are not known to infect or cause illness in people. The spread of the virus depends on large respiratory droplets, that is particles over 5 micron in diameter, that are expelled from the respiratory tract during coughing and sneezing. And particles do not remain suspended in the air, and close contact above 3 feet is required for transmission. Now, direct contact with respiratory droplets or secretions, followed by touching the nose or mouth, can also transmit the disease or the virus. The incubation period of this virus is about 1 to 4 days, with an average of 2 days, and adults may be able to infect adults beginning 1 day before getting symptoms and up to 7 days after getting sick. Children can be infectious for up to 10 days, and young children can shed virus for up to 6 days before their illness onset. And severely immunocompromised people can shed virus for weeks or months. Now, the problem with this virus is that the virus mutates and new strain emerges, causes a new round of infections, and so on. So each year, new flu vaccines are needed for the current virus. The replication cycle of influenza is illustrated below. Now, this illustrates many of the features of a typical animal cell virus, such as all virus begins the viral cycle with adhesion or attachment of the viral particle to specific receptors on the target cell surface. This adhesion triggers uptake or endocytosis of the virion by the target cell, followed by uncoating and release of the genetic material into the host cell cytoplasm, and subsequently in the nucleus for this case. Now, this genetic material takes over the machinery of the host cells to make more proteins and replicate the viral genome. After all the viral genetic and protein materials are synthesized, it will begin the assembly process of new virions on the host cell membrane, and they're eventually budding from the host cells and taking the host cells' phospholipid, modified with virus proteins, and with them as their viral envelope. There are 2 different surface glycoproteins on the influenza virus surface. HA, or hemagglutinin, bind to sialic acids on human cell surface. It facilitates entry of virus into cell via receptor-mediated endocytosis. Na, or neuroaminidase, on the other hand, facilitates exits of virion from host cells at the end of the viral cycle through exocytosis. Protective immunity against influenza involves both the innate and adaptive immune mechanisms against viruses, including cellular and humoral responses, CD8 T cells, NK cells, and interferons kills virally infected cells. However, the primary protection against influenza is neutralizing antibodies directly against the trimeric HA and NA glycoproteins. Now, if an individual received a seasonal flu vaccine, they would have antibodies that may be effective in neutralizing the virus before it can infect cells. I said antibodies may be effective in neutralizing the virus because the seasonal flu vaccine is always the best educated guess. And WHO scientists predict the most likely virus strain every year, and the vaccine manufacturers based on that recommendation to prepare the vaccine for that particular season. The guessing game is not always correct because of antigenic variation in the influenza virus. The variation is because influenza is an RNA virus, and RNA replicates in a relatively error-prone fashion, and there are no machinery like we have in place to correct and repair that mistakes. And therefore, structural changes of HA and NA translate into altered antigenic properties and referred to as antigenic variation and antigenic drift. In the figures, person P is infected or carrier that can spread virus to person Q, and virus mutates in the process of replication in person Q, and that changes the structure of the glycoprotein, and that changes the antigenic properties. And next, person P now can be reinfected with the mutated strain. Now, in fact, it happens every winter, and this is referred to antigenic drift, and it is responsible for a cycle of mild flu epidemics every year. Antigenic drift is the reason behind the slight antigenic variations in the seasonal flu virus. On the other hand, an antigenic shift is a genetic recombination event that allows virion to evade host cells in multiple species. Now, in antigenic shift, the idea here is that a third party, such as a pig, is infected simultaneously with human and avian or bird flu strains. The two different strains of the viral RNA mixed or have a recombination event in the same pig cell. At this point, the change is not just a point mutation, but a drastic change that contains part of the genome from avian and part from human. Now, when the virus infects humans again, the immune system really doesn't know what to do with that because it becomes a brand new virus, sort of. Now, this mechanism could be a reason for what happened in late 2019, but the investigation is still ongoing with much debate. Besides antigen variations found in bacteria and viruses, our adaptive immune system also has two basic mechanisms that make our immune response not as effective. Now, it turns out IgG can suppress naive B cells activation during secondary immune responses. Now, during a primary immune response, that means encountering an antigen for the first time, a pathogen binding to the antigen receptor of a naive B cell delivers a signal that activates the cells to become an antibody-producing plasma cell, shown in the left panel. However, during a secondary response, in which the antigen receptor and the Fc receptor, Fc gamma Out2B1 on a naive B cell can be cross-linked by a pathogen coded with the IgG. This delivers a negative signal that prevents the activation of the cell, shown in the middle panel. This can help prevent new B cells from generating low affinity IgM and ensure an efficient response from memory cells to produce high affinity IgG. Now, in contrast, a similar cross-linking between the antigen receptor and Fc gamma R2 B1 on a memory B cell activates the cells to become an antibody-producing plasma cell, shown in the right panel. Now, this overall mechanism has a major disadvantage since naive B cells activations are suppressed. This may prevent primary response against new strains with slightly different epitopes. The last mechanism that is responsible for immune failure is called original antigenic sin or immune imprinting. It happens when a highly mutable viruses such as influenza gradually escape from immunological memory without stimulating a compensatory immune response. And let's find out what it means. OAS is a type of immune imprinting or a type of immunological memory. The first time our body is exposed to a pathogen, for example the flu virus, our adaptive immune system will react and remember the specifics of this virus, like an imprint. The next time we encounter the flu virus, our adaptive immune system remembers this imprint and quickly produces antibodies to neutralize it. But there is a big catch because there are multiple different strains of flu viruses. When we are young, we develop an imprint of the most frequent flu virus strain at the time. When we are infected later in life, our immune system goes back to the imprint we first learned. If this strain is similar to the imprinted strain, we would have no problem clearing this virus. But if it is a different strain, the antibody generated would not work as well and the risk of infection would increase. In summary, a massive event of immune failure in a population could lead to a pandemic or an epidemic. Our generation has lived through such event since March 2020. This leads to our next lecture, or perhaps the most anticipated lecture in this series, where we will talk about how vaccines stimulate immunity to prevent and lessen the chance of immune failure before an infection. So stay tuned, and I will see you next time. Bye.